Thank you very much for the kind introduction, Julie, and I want to welcome each and every one of you, members of the broad SLHD family, distinguished guests, visitors, benefactors and donors, and of course the Gadigal people of the Eora Nation and their elders past and present, who I provide my respects to, as we all do, I know. Today I want to try and convince you of a single compelling idea that has obsessed me for at least a generation. The idea that we might be able to use the very genes in the cells in our body as medicines. This is an idea, as I say, that has compelled me over the last 25 years to pursue this as a means of adding a new dimension to medical therapeutics. You've heard already today about disruptive technologies, self-driving cars, for example, and I'll be mentioning another kind of car as just one of 4 examples that I want to use to convince you that gene therapy and cell therapy, as well as regenerative medicine, is here now today. Underpinning everything that I talk about today will be an understanding of genetics. And as a very first start, I don't know how many of you were paying attention during your high school days, learning about science, learning about the evolution and the development of genetics, but these are the 2 fathers of genetics, and I want to just do a quick experiment. 1 hand up if you know either one of them, 2 hands up if you know both of them. Let's go. Obviously everybody's asleep after lunch. Aren't that many 2-handed? On the left-hand side, we've of course got the pea priest, Gregor Mendel, who's so 1860s these days, with his friend Charles Darwin around about the same time, who's saying, "You got that right, bro." See, he was a priest. That's my only joke, being a nerd. That's all I've got to say about that. They didn't even know the term DNA, which of course everybody in this room knows stands for DNA. Very good. But it is the foundation of everything that I will be talking about today. Because when you reflect on what we now understand, the double helix, the fact that all of our cells contain the information stored in this chemical DNA that gives rise to what we are. And what we find when we look at the DNA is that all of us are 0.001% different. That's the only difference between us. The difference between us and monkeys is only a few percent. Us and mice, maybe 10%. Us and worms and plants, maybe 40–50%. Life is a uniform celebration. But those differences are like the lottery. The lottery of the DNA from the mother and the father, and when a mummy loves a daddy, you all know what happens. A new life is filled with a combination of the 2 genetic legacies from the mother and the father. Some people refer to that as a genetic lottery, and you might see it as a genetic dealing of the cards, because incorporated into that genetic information, which is stored as a sequence of so-called chemical bases, C, A, G, and T, all sequenced 3 billion of them in each one of your cells, storing the informational content of what you do and what you are. That is what makes us what we are, but hidden in that is also the possibility of disease predisposition. And that's what we now understand about molecular genetics and the field of genetics. It is a diagnostic and a prognostic specialty of medicine that's changing the world. And at this point, I want to suck up to Julie McCrossan as the chairperson of the debate, who I honour and respect enormously because she will be influencing whether or not we win in the subsequent debate to my talk. Nonetheless, this genetic payload stores in it the information that might also predispose us to genetic diseases and sometimes cancer. Some 4,000 human genetic diseases have been identified, and that information passed on from the parents to their descendants is stored in the DNA sequence. The idea that I want to convince you of today is that we can use that information not just to make a diagnosis, not just to suggest what might happen in an individual, or even potentially select embryos using IVF technology, but actually use the information itself as a therapeutic. In other words, put the correct version of the DNA back into the body. By doing so, we add a living therapy. In other words, because it continues to survive in the cells as they divide. It is a compellingly simple complex. If you're born with an abnormal gene, let's just put back the normal one, doctor, and prevent all of these problems. It's paradigm shifting and disruptive and no different to something like Uber or self-driving cars, as you've heard about today, simply because it is a new way of doing medicine. It's not like a tablet that you have to keep taking. It's not like radiotherapy. It's not like surgery. It's something new. And what I will convince you of, I hope today, is that we're making the first steps towards establishing this as a proven therapeutic component of what we have able to treat our patients with. There are a lot of words used in this area. Gene therapy, cell therapy, immunotherapies, and a bunch of others that decorate our portfolio of words that we use. You'll even see personalised medicine there, for which the affirmative I will be defending in just a few minutes' time. But the fundamental idea is that we get cells, which are the building blocks of all of our tissues in our body, and those cells within their nucleus store the DNA, the genetic information which may store disease-causing genes. We need to get DNA into those cells to correct them. And typically we use viruses to ferry new genetic information into those cells. So the idea of gene therapy then is to join genetic therapy to a cellular therapy and then make a clinical benefit. And the technological challenges have been enormous to achieve that, and it indeed has taken some 30 years. I often get asked, well, John, okay, that all sounds theoretically fine, but which genes are you going to choose? There are some 20,000 genes stored in our 3 billion Cs, As, Gs, and Ts, and the gene is specific to the particular disease. So if you're born with an abnormality of one particular gene, well, why not put back the normal version of that gene and see if we can get a therapeutic benefit? Where do we get the genes from? Well, they can be chemically synthesized now. We can take them out of other cells, manipulate them, and then using a virus typically as a vector or a ferry to put the genetic material into cells, that's the way we achieve this. So, when we ask the question, which genes, I'll give you now 4 examples, clinical examples of where we're headed. The first one is called CAR T cell therapy. It's a form of immunotherapy, and it uses the body's own immune cells as a treatment for one's own leukemia or lymphoma. Oftentimes when I'm breaking the bad news to patients that they have a disease like cancer or leukemia or lymphoma, they look at me and say, but doctor, my immune system, what's happened? It must be weak, it must be failing. Why isn't my body rejecting this horrible cancer? To which the answer is, somehow the cancer has evolved or mutated in your body to prevent your own immune system from attacking it. So, here's the idea. Reprogram your own immune cells in your own body so that they now attack the cancer. And that's exactly what's been done over the last decade using this gene and cell-based technology specifically in diseases like acute lymphoblastic leukemia and diffuse large B-cell lymphoma in adults and children. And if you don't believe me in anything I say today, believe the market. Because last year, when this announcement was made and the approvals were given for this technology in the United States, one of the 3 companies that were developing this technology was purchased within 2 days of the announcement of approval by the FDA for $12 billion US. So what's involved? This is basically a crash course in gene and cell technologies, personalised medicine for lymphoma and leukemia. A patient has their own immune cells taken out of their body from a vein, these sleepy immune cells. They're then manipulated in a space station-like laboratory, which I'll show you pictures of in a while. And that manipulation requires genetic modification and expansion in the laboratory in a safe and sterile manner. Those cells are then validated and tested to make sure that they're safe, sterile, and correct. And then a patient receives, with some chemotherapy, a dose of their own cells that have been genetically reprogrammed to attack a marker on the surface of their cells. This is a real picture, a fly around of these genetically reprogrammed T cells, so-called CAR T cells, that have been taught now to target a protein on the surface of leukemia cells, in this case coating beads. And you can see how these cells are wrapping themselves around the beads because they're engulfing them and attacking them and trying to destroy them. These cells have been referred to as serial killers because just one of these immune cells is capable of eradicating the entire body of a leukemia or a lymphoma. One single cell. They're capable of 10,000 or 100,000-fold proliferation and expansion. Just like when you get an infection, your immune system turns on and then turns off. And so these cells then, once they're replaced back in your body, can not only kill a leukemia or a lymphoma, but then they return to a state where they're monitoring things in case it comes back. Again, if you don't believe me, believe the results of clinical trials which have proven that this works in two diseases, acute lymphoblastic leukemia of childhood and diffuse large B-cell lymphoma. And I can tell you that this picture tells 1,000 words. This happy dog has been This young woman, Emily Whitehead, became the poster girl of immunotherapies in the United States. She met with Obama and she lobbied the Food and Drug Administration. She was one of the very first people on the planet to receive immunotherapies. She had advanced leukemia and was literally within weeks of death. And there she is now 6 years, in fact, this is a year old, 6 years after the single injection of this living immunotherapy of her own genetically modified cells that attack the leukemia and lymphoma. A paradigm shifting and revolutionary result. So, the two approved treatments in the United States I've made mention of are listed here. They were approved in the middle of last year in the United States, and we await the likelihood of them being approved in Australia because we at Royal Prince Alfred Hospital were part of the industry-sponsored study by Novartis that has contributed to these discoveries. You can see the names of them there, and the drug companies, of course, name them so that they can easily be pronounced. Tisagenlic, and the other one, Acicab. You can't pronounce them, they make it that way, but you can see Kymriah and Yescarta are the two brand names which everybody can readily pronounce. So the bottom line is, this presents a new way of delivering therapeutics to humans. Everyone understands chemotherapy, radiotherapy, surgery, some biologicals like recombinant antibodies and some targeted therapies, but this is a new way of delivering therapeutics using the body's own cells, reprogramming them with genes. The advantages are compelling. It's targeted against the specific leukemia, and it is a living therapy, as I've explained earlier. There are disadvantages, and there's research that's being undertaken to address every one of these possible disadvantages. You need to defer the treatment because the cells need to be manufactured in a laboratory. There are significant side effects in a small proportion, about 20% of patients, that need to be well managed by intensive care units sometimes. And of course, sometimes these cells are capable of attacking one's own normal residual immune cells, and that requires immune support. It raises, of course, a massive other question, which is that of reimbursement. Everyone has heard the idea of drug companies loving inventing medicines that one needs to take for the rest of your life. When did you last get a guarantee from your oncologist who said, look, you can have your money back if I don't cure your cancer? That's something that's so shocking that you haven't even thought of that possibility. The reimbursement models of these incredibly expensive therapies that will be probably priced at over $500,000 for a single injection are mitigated by the fact that there are new reimbursement models that have previously not been even considered called adaptive reimbursement, where, for example, in the United States, the company says, if you haven't responded within a month, you don't need to pay. That's how confident they are of this kind of technology, and indeed integrating this technology with all of the current armamentarium of treatments is something that we'll be doing in the coming years. We hope that as a consequence of this new technology, example number one, that you will be seeing newspaper articles like this one, which will be generically applicable to a much broader range of cancers. This is specific to those two types that I've mentioned there. There's a whole pipeline of new ones on the way, and indeed we at Royal Prince Alfred Hospital will be exploring this in a number of solid tumours, which is the next big thing in this area. A second example that I really want to highlight is one that I haven't been involved in, but one that I've been watching, in a sense, for many years because it's been developed by a company that we've collaborated with for 20 years. If you'd asked me 25 years ago, could I have imagined a gene therapy to cure blindness? I would have said, like, how? It's not possible. This is imaginary stuff. But doctors in Philadelphia, led by Jean Bennett and Kathy High, have developed a technology that does exactly that in a disease called Leber's congenital amaurosis. Babies are born with a defect in a gene called RP65, and they become progressively blind over the first few years of life. There is no treatment for this disease. They had to develop technologies to inject the gene therapy at the back of the eye in the retina where this disease manifests itself, and then develop new electrophysiological testing and even little mazes to show whether or not these patients who were treated in childhood were able to avoid the complications of blindness or indeed cure their blindness. This is a young man who is clinically blind, standing in front of a maze. One of his eyes are covered now. It's the blind eye before he's received the gene therapy. You can see that as he attempts to walk the maze, he even doesn't see this big stop sign hanging right in front of him. He can't make his way forward. He's trying, but there he is. I can't move forward. Whoops, he bangs into it and can't make progress, as you can see. The design of the study was so clever because at the start of the study, the decision was made to inject only one eye first with the gene therapy and leave the other one intact as a blind eye. And so now in the second video you'll see the gene therapy receiving eye is the one that is open and the blind eye is still blind. And now he can walk quite confidently and has essentially been cured of his blindness, and of course the second eye was then safely injected. This treatment that was in clinical development for 20 years was approved last year in the United States of America, and we expect it will be approved in the near future in Australia, and we hope we'll be delivering it for individuals with blindness near in our facility at Royal Prince Alfred Hospital, and certainly at the Sydney Eye Hospital as well, and other hospitals as this technically demanding technology becomes applicable. The second last example that I want to highlight is that of haemophilia, which I've been passionate about for nearly 20 years plus. This is a disease of bleeding, as many of you would know. There are different types of it, haemophilia A and haemophilia B. We first focused on haemophilia B and used a virus that is safe and innocuous to ferry in the clotting factor gene that these people lack. You would all know the story of Rasputin and the fall of the Russian Empire because the Tsarevich Alexis had haemophilia. You would also know that the royal houses of Europe carried this haemophilia B gene and indeed affected human history in so many ways. What we have developed over 20 years in collaboration with the Children's Hospital of Philadelphia and now Spark Therapeutics is a one-time shot in a peripheral vein of gene therapy. And if you can imagine what a difference this would make if it were to work, let me remind you that individuals who suffer from this disease at the best need to inject themselves every few days with recombinant factor concentrates or clotting factors purified from other generous blood donors. And if you work it out on a lifetime basis, they need to inject themselves 12,000 times throughout their lives to prevent bleeding. We're talking here about a single injection, and let's see how we went. If you look at a plot of months after the gene therapy, and you look at the factor 9 level in the y-axis there, I haven't indulged myself very much with graphs, but I'm so proud of this one I had to share it with you. This one graph I hope will show you that if you have haemophilia, you always have severe, less than 1% levels of factor 9. And so unless you inject yourself with concentrates that go up and down, you will stay very low throughout. This is the results of the first 10, 11 patients that we treated in this clinical trial that was recently published. All of the patients were, for all intents and purposes, cured. following a single injection of the gene therapy that ferried its way into the liver, which is where all of us, I hope, are making our normal clotting factors, and then started using one's own liver as a factory as nature originally intended. That's the technical results. This is the only other one. There's the before and after. The 10 patients on the left are individually themselves controlled. You can see the number of annualized bleeding and the annualized factor that they required in the year prior. Then there's the gene therapy in the middle, and that's what they needed to inject after the gene therapy. So a 99% reduction, an amazing change in the quality of life of these individuals, but also a potential massive cost saving. This is not yet approved anywhere in the world. We hope it will be approved in the United States soon, and we hope it will be approved soon after in Australia. We did see a couple of immune responses to the coat of the viral vector, and that required a few months of steroids. So in 2 of the 11 patients, one of whom was our own, we required 3 months of steroids, but that was the only consequence of the gene therapy that was negative that we observed. And I can tell you that Mark Lee, one of our patients who allowed himself to be photographed and participated in our announcement of this discovery, was absolutely thrilled. He's a mechanic, salt of the earth, and came up with his family to celebrate the fact that he is now independent of clotting factor concentrates and feels fantastic. The last example I will give you is a larger one, because the examples that I've given to date are relatively rare diseases. This one is thalassemia. It's been described for probably since the beginning of humanity, but because of its Greek origins, thalos from the sea, emia, blood. These people from the sea had a weak blood, a disease of chronic anemia from birth. So in severe transfusion-dependent thalassemia, you require blood transfusions if they're available, and because you're having regular blood transfusions, you need to remove the excess iron from your body, which means chronic removal of iron using tablets or injections. We have co-evolved with the malaria parasite, it turns out, so that in equatorial regions on the planet, there is a high incidence of thalassemia. 5% of us carry 1 copy of the thalassemia gene, and if we get 2 from our parents, 1 from each, then we can have severe transfusion-dependent thalassemia. You get the hang of how this works now. Basically, we took individuals this time not to take out their immune cells, but to take out their bone marrow stem cells, their blood-forming stem cells. We coax them out of the bone marrow using drugs, we siphon them off using a special machine, and then we genetically modify them outside of the body and then reinfuse those cells with chemotherapy. These are the results, and essentially what I want to say is that more than half of the patients that received this form of gene therapy of blood-forming stem cells were essentially cured. The before and after was that they needed transfusions every few weeks in their life. The after was they never needed any more transfusions in half of the patients, and everyone received a benefit of around about a 2/3 reduction in the number of transfusions and the frequency of transfusions. And this is a picture that actually brings tears to my eyes usually, because it's the last time that one of our patients ever needed a blood transfusion in his life. After this treatment, his life was completely changed. He never needs a blood transfusion after that. In order to achieve these advances, a number of technical advances have had to have been achieved. People often ask me, as I've been banging on about this technology for the last 20 years, how is it that it's taken so long? And the investment and the time that Sydney Local Health District, Royal Prince Alfred Hospital and the chief executives of the hospital have invested in what was unproven up until recent years is a testament to the excellence of our institution and the commitment to research. Those technical advances have included gene transfer efficiency, which had to be overcome using the viral systems that I described, a very strong commitment to safety and regulatory experience, as well as compliance and quality assurance, and I take my hat off to all the individuals in this room who have committed themselves to those specific aspects of their career, and of course breakthroughs that are specific to different diseases. Think of the blindness one, the surgical expertise and technology required to develop just to be able to make an injection at the back of the eye. Those technical advances are taking us in a certain direction, because I think you'd have to have your head in the sand not to have heard of the new ideas of gene editing and gene scissors that actually allow us not just to add genes back, that I've been describing up until now, but actually changing any abnormality in the genes themselves. So gene editing, gene scissors, or gene correction. And I call myself a hematologist. I still practice as a blood doctor, but really for many years I think I've been in evolution to a cell and gene therapist. One day your doctor might walk into the room, being a gene scissor doctor, look a little bit like this perhaps. The last topic that I want to just touch on is regenerative medicine, which offers hope to many people growing tissues. You heard the exciting work of skin and the burns unit this morning, and so many of the robotic surgery and the academic surgical unit are committing themselves to making organs outside of the body as we are. But there is a dark side. And that dark side is the proliferation of clinics that are selling unproven cell therapies. And I've spent many, many, many years writing, talking to consumers, talking to government about the problem that is the false hope that is offered by some clinics that are purporting to treat diseases for which there is no evidence whatsoever that cells are doing that. And you would know, many of you, that in a couple of weeks' time the TGA is cracking down on that after some 7 years of having a loophole that has allowed these clinics to proliferate in so many parts of the world. In familiar parts such as, for example, Russia, the Philippines, Southeast Asia, but this proliferation is something that we were very concerned about a few years ago. And so with a group of medical students, I actually set to try and document the proliferation of these clinics. So what we did was we went online and we rigorously searched about direct-to-consumer marketing of all of these clinics that were advertising stem cells online. And we asked the question, where are these clinics and who are they advertising to? These were all English-speaking clinics, and then we mapped them geographically to where the clinics were located. And what was shocking to many people was that the highest incidence of clinics per country was in the old US of A. A massive contribution in Florida and in California in particular, but an incredible proliferation of these clinics, hundreds and hundreds of them. But then we did a recalculation, and I'm going to change this map not at the number of clinics per country, but the number of clinics, as you'll see here, per capita. And the darkest, brownest, will you forgive me, the shittiest of the countries that had the highest number of clinics per capita was Australia. No. Yes, Julie, I know. Towards 100 clinics directly marketing to Australians, and these are unproven. Which begs the question, if you're going to spend your hard-earned dollars on something, how do you know whether it's proven or unproven? You have to have a very rigorous system, which we have the proudest health system in Australia, and I'd like to say Royal Prince Alfred is the proudest of that health system. But here you can see what's proven and unproven. And if you go to a cocktail party tonight and you're standing over a beer or a champagne or a big drink of gin and tonic, you might be asking yourself the question, well, I've got sore joints, I've got some unmet needs in my health. What should I be doing? I'm hearing it's advertised online, I'm hearing it on the radio, what should I do? Well, if you as health professionals and people who are informed in this area are confused, just imagine how people who are less sophisticated than you are must be confused about these alternatives. It's a real problem, it's here and now. So here is then a reality check of what is approved in the cell and gene therapy area in the United States right now. This is just the gene therapy approved products, and I've touched on pretty much all of them except one, which is the Provenge for prostate cancer. All of these technologies currently need to be manufactured in highly sophisticated laboratories. They require advanced sites. And this is a picture of the Cell and Molecular Therapies department at Royal Prince Alfred Hospital. Not only do you need an advanced laboratory, but you need PhD scientists, hospital scientists, dedicated quality managers, clinical trials staff, the whole gamut of services that is required to develop an advanced technology like this. Royal Prince Alfred has committed itself to developing this technology, and what I've shown you today are some examples of that. So gene therapy, I would like to submit to you, is here and now. there's still a long way to go before it's applicable to many different diseases beyond the few that I've mentioned, and they all need to be studied further to how to integrate these technologies in the broader existing armamentarium that we have available to us. But so far, we can probably give tentative ticks to things like thalassemia, blindness, haemophilia, and indeed leukemia and lymphoma of specific types, which then I hope it might convince you that we may soon be able to modify some of the genes you are dealt with in terms of causing severe human diseases. And so this is a conclusion of I'm a Gene in a Bottle Baby, and I asked Julie to perhaps join me for a little bit of a jig, if that's available. Please come up.
I have the rhythm of a stick, John.
Oh,
but
really?
Can you play John's music? Because he was so shocked that I didn't recognise the singer.
Didn't recognise it.
Can you play it?
I thought I was, but it doesn't seem to be going. Never mind.
Look, I didn't know it anyway. Ladies and gentlemen, would you agree that that was a bloody good presentation?